It's a great pleasure to be here today. I'm especially excited that Leland is here with us to give a talk later at 11 o'clock. So I'm sure many of us at the BERT are big fans of UMAP. But before we get to that, in this talk, I will tell you about some of my recent work on single cell data visualization and sketching that are closely related to today's theme on data embedding. And my hope is that this will also provide some biological context for Leland's talk later and also for some of the following discussions. So as you all know, one of the ways in which single-cell transcriptomics has been transforming biology is by allowing researchers to easily grasp and explore the transcriptomic landscapes spanned by single cells in living organisms. And these landscapes, which are often explored through visualizations like the ones that I'm showing you on the slide, reveal many useful biological insights, such as the presence of different cell types or transcriptional states, or the structure of more complex patterns like developmental trajectories. However, an important fact that is often overlooked is that when we think about these data sets based on these geometric representations, we're actually working with artificial representations of these structures. And in reality, these cells live in a very high dimensional space with thousands or tens of thousands of dimensions. So this naturally leads to the concern that our perception of this landscape may be misleading. And here I want to draw an analogy to cartography. This is a world map that we're all used to based on the Mercator projection, which works by first projecting the Earth onto a cylinder and then rolling out the cylinder into a 2D plane. And because this projection summarizes a 3D surface in 2D, it suffers from distortion where it stretches out the lands as we get closer to the two poles. In fact, if we were to rescale the map to reflect the true size of each country, then this is the kind of map that we would actually get. Social scientists have argued that the Mercator projection leads to implicit biases in how we think about different countries around the world, which is unfortunate. The key question we ask in this work is whether we also have these kinds of biases in single cell data analysis. And as I will show later in my talk, this is indeed the case. A particular aspect of transcriptomic landscapes that we focus on in this work is that of spatial density. So if we imagine a complex gene expression structure in high dimensional space, there will be regions that are densely populated, and there will also be regions that are more spread out and sparsely sampled from. And biologically speaking, these differences in density could reflect differences in transcriptomic variability, where the sparse regions corresponds to highly variable cellular states, and the dense regions represent more tightly regulated ones. In the rest of the talk, I will show that many existing analysis tools do not take this density information into account, which in turn limit our understanding of these landscapes. And I will also explain how we devise new strategies to overcome this limitation. So along these lines, I will present two of our recent works that aim to introduce density awareness into single cell analysis. The first is about how to visualize single cell data while faithfully capturing density landscape. And the second is about how to sketch or downsample a large single cell data while accounting for the density bias in sampling. So let me first start with the visualization method. As I mentioned, data visualization is a standard step in single cell data analysis. And just so we're all on the same page, the goal here is to represent the gene expression landscape in 2D or 3D, where each point represents a single cell, and cells with similar gene expression patterns are located closer to each other. These visualizations are useful not only because they make salient structures of the data immediately clear to the researcher, but also because they allow new kinds of analytic approaches downstream. So for instance, additional data can be projected onto these visualizations to convey different properties about the landscapes. So the figure in the bottom left corner overlays the expression level of individual genes onto the visualization to reveal interesting patterns of gene expression distribution. So two of the most popular tools for visualizing single cells, as I'm sure many of you are aware of, are t-SNE and UMAP. And unlike traditional approaches like PCA, which finds a linear projection of the data down to a low dimensional space, t-SNE and UMAP learn a nonlinear embedding, which turns out to be better suited for capturing complex manifolds that are commonly encountered in single cell genomics. And these have been widely successful in this domain, as you know, and it's almost impossible to find a paper in this field that doesn't have a figure like this in it. So here's how t-SNE and UMAP work at a high level. So we're starting with a gene expression matrix from a single-cell RNA-seq dataset. These methods first construct a type of pairwise distance matrix for the cells based on their gene expression patterns. And this intuitively captures the structural patterns in the data. And it is common to use things like k-nearest neighbor graph to make sure that this matrix is sparse, which is easier to work with. And this can be thought of as putting emphasis on the local structures in the dataset. And these methods apply a similar process to the embedding to compute a pairwise distance matrix based on the embedding coordinates. And these coordinates are then in turn optimized, iteratively optimized to minimize difference between the two distance matrices. And this encourages the embedding to accurately reflect the structure of the data in the original space. The main differences between t-SNE and UMAP lie in how they define the distance matrices and the way in which they measure the difference between them. Here, I do want to point out one key concept in both these methods that make them especially effective for capturing complex structures. So we refer to this notion as adaptive length scale. So let's say we have these two groups of points in high dimensional space. And depending on the length scale at which we view this data set, we end up with a different conclusion as to what kind of structure this represents. So if we choose a small length scale, then the example on the left would look like a cluster, whereas the one on the right would look like an unrelated set of points. On the other hand, if we choose a large length scale, then the one on the right might start to look like a coherent cluster, but then we might end up lumping up the one on the left with other local structures and therefore missing information about local structure. So in both t-SNE and UMAP, this issue is addressed by choosing the length scale adaptively for each point in the dataset. This is done by looking at a small number of nearest neighbors of each point and finding a length scale that makes each of these local neighborhoods to appear roughly comparable in size across the dataset, shrinking and stretching the space across the manifold as needed. And this turns out to be a very effective technique for capturing data manifolds, complex data manifolds in various domains. However, there is an unfortunate side effect to this technique. The problem is that density information is now lost. So here I am showing you an example of 6 Gaussian point clouds with different variances, and each cloud contains the same number of data points. So if we were to apply the adaptive length scale idea to this setting, then we would end up choosing a length scale for each cluster that roughly corresponds to the size of the cluster, which would make all of these appear roughly the same in size as a result. And this is indeed what we see if we apply t-SNE or UMAP to this data set, as you can see in these plots. And in fact, this is a more general problem that shows up in a range of different settings. If we have a dynamic trajectory of a point cloud that gradually increases in variance, existing tools would discard information about this change in variance and end up with a uniform trajectory like this. And if we have a set of equal sized clusters with different sampling rates, then the sparse clusters tend to appear much smaller in the existing visualizations because larger length scales are chosen for these clusters. And similarly, if we have a continuous structure with varying sampling rate, then the densely sampled regions tend to be more stretched out, which distorts our perception of the underlying shape of these data. So I will show later that these behaviors can lead to misleading visualizations for single cell data sets. So how do we fix this problem? Our main idea here was to augment the objective function of each algorithm to incorporate information about density. So recall that both t-SNE and UMAP optimizes an objective of this form, which maximizes the agreement between the original pairwise distances and those in the embedding. In our methods, we add an additional term that measures the correlation between local densities in the original space and those in the embedding. In this way, we can modify the behavior of these algorithms so that in addition to capturing local structural patterns, which these methods are already very good at, we can also encourage them to faithfully represent density differences in the data across different regions in it. Of course, we need a weighting term here, shown here as lambda, which controls the relative importance between these two goals of the algorithm. Now, the key challenge for us in making this idea really work in practice was to design a good notion of local density that can be efficiently optimized in conjunction with the existing objective term. So first of all, we need a formula that is differentiable with respect to the embedding coordinates, so we can optimize them using gradient approaches. And on top of that, we also want the gradients that we compute to have an efficient form so that they can be easily computed during optimization. So figuring this out was like one of the main parts of this work that took many months for us to get right. I won't go into too much detail here, but here's a high-level intuition of what we did. So we defined what we call local radius as our measure of local density. So for each point, remember that we already have an adaptively chosen length scale from the original methods, here shown as sigma I. And along with it, we also have normalized distances to the nearest neighbors from each point, which are scaled accordingly by the chosen length scale. And these distances, which we can transform into a measure of connection strength, can be taken together to be viewed as defining a smooth probability distribution over the neighbors of each of the chosen point in the data set. And because of adaptive length scale, this distribution roughly corresponds to a notion of a smooth neighborhood, equal size neighborhood around each point in the data set. Now, given this, we then define local radius as the average or expected distance to nearby points within this weighted neighborhood. So note that this naturally captures the notion of local density. So dense regions will have small average distances and sparse regions will have large average distances. And also, this term is differentiable and maximally reuses the quantities that are already computed by the underlying methods, including the length scale and the normalized local distances that we use in our weighted average. So as a result, our design of this local density measure led to an objective term that can be easily optimized with minimal computational overhead. So to summarize, here is an overview of our density preserving visualization methods.
I just want to slow you down for a second. How do you make it smooth? It's like, I thought you were going to say something more like a kernel density estimator, but that's not exactly what you're doing, is it?
Right, it's pretty close to that. The sense in which I said this is smooth is because we're putting this smooth weighting term over all of the neighbors in the neighborhood instead of choosing a fixed radius and then cutting it off, right? So,
oh, yes, I see. So there is a kernel. Yeah. Okay, good.
Yes, yes, exactly.
Okay, great. Thanks.
Okay, so this is the overview of our methods, dense-SNE and dense-map, which augment t-SNE and umap respectively. Existing methods, as I mentioned, approximate the manifold using k-nearest neighbor graph, and then they learn an embedding that tries to preserve these local distances. And because of adaptive length scale, the result is a visualization where density information is lost. For each of these methods, we leverage the local distances that were already computed by the underlying method to additionally calculate local radius for each of the points in the data set. Then we can jointly optimize the original objective now with an additional term that measures the correlation in local radius between the original space and in the embedding. And this produces a visualization that conveys the same local structures captured by the original methods, but additionally with extra information about density.
So there's a little tuning parameter, right? Because you're kind of compromising between the distance, the adaptive distance preservation and the density preservation?
Exactly. So if you set that weight to 0, then we reduce to the original methods, and if you increase it really high, then it will start to, sacrifice some of the clustering patterns or what have you. Yeah.
Okay, because I imagine that at some point there's a tension between this density preservation and the kind of adaptive nature that actually works so well to do the to preserve other kinds of structures.
Yeah, there is a trade-off, and
the high to low dimensional kind of thing is right. Sorry. So
from what we've seen, there's a sweet spot in terms of where the right parameter setting is. I mean, that sweet spot was actually different for t-SNE, between t-SNE and umap. But all the results I'll show you later are using the same parameter choice for all the data sets, and these seem to work well.
You were able to find some robustness there, even with the wide range of different input parameters for the data. I mean, data kind of configuration.
That's right.
Nice.
Okay, so here are some examples. So when we apply our new methods to the examples that I showed you earlier, we observe across the board that the shape of the underlying data set is more accurately captured now. So in this example of 6 Gaussian point clouds of varying variances, now the dense-SNE and dense-map images more accurately reflect the true sizes of these clusters. And the same is true for this trajectory data, where now we can see this expansion pattern in our images. And for the case where we have 6 clusters of the same size but different sampling rate, now we convey the information that all these clusters have similar size, and we end up fixing this problem where sparse clusters appear smaller in the images. And lastly, in this example, flying carpet example, which is what we call them, we fix this distortion issue of dense regions being stretched out more than the others, and now we have an image that more accurately reflects the underlying shape of the data.
This is Luca. I have a question here.
Sure.
So here you are showing original data that is in 2D and embedding that is in 2D. And so
it's actually high dimensional.
What is high dimensional? The original data? The
original data is high dimensional. I am just showing you like a t-SNE plot just for visualization purposes, but these clouds are sitting in high dimensional space.
Okay, cool. Thank you.
Except for this carpet example, which is a 2D object that we embedded in high dimensional space.
Cool. But maybe I can ask the question anyway. That is, as Alex was mentioning, there is probably a competition between those two terms. And my intuition is that that competition will be more severe the largest the difference between the original dimension and the embedding dimension. While there is no tension at all, right? If the original dimension and the embedding dimension are the same, you can embed keeping both local distances and density.
Yeah, that's an interesting idea. Yeah.
Yeah. So I'm wondering if, there is you have any intuition about how can you, what's the tension between those two terms as a function of the dimension of the original space?
So I would have to think more about this issue, but my intuition would be that the way in which the differences in the original dimensionality affect the behavior of the algorithm, I think the impact on the original methods would be similar to the density term that we introduced as well, because ultimately the density measures that we calculate are also based on the k-nearest neighbor representation of the data set. So in some sense, like all of the distortions that result from reducing the dimensionality already happen in that structure, like the k-nearest neighbor graph that we base these methods on. So in some sense, maybe this wouldn't affect the weighting term that balances these two terms, but it's definitely true that this would affect the behavior of the algorithm overall. I'm just not sure about the trade-off between the two.
Thank you.
Okay, so so in the following slides, I will try to show you some case studies of how these methods actually work on real single cell data to give you more intuition about what they actually do in realistic settings. So the first one that we tested was human peripheral blood data set from Jung et al. And 10x Genomics. And this data set included around 68,000 cells. And when we applied both DenseMap and UMAP, we were immediately able to see some clear differences in how relative sizes of different clusters were conveyed in the images. There were some noticeable differences over here in the T cell and B cell populations, but overall, the largest difference was observed in these clusters representing monocytes and dendritic cells. So we zoomed into this area for further analysis. So these two images are zoomed in versions of the regions that I highlighted in the previous slide. With DenseMap on the top and UMAP on the bottom, we color these cells according to their cell type. So the orange corresponds to monocytes, purple corresponds to dendritic cells. We also labeled lower level classification of these cell types using the information from this paper by Leland et al. So there are a couple of things to note here. So there's a striking difference between the sparsity of the CD14 monocytes and the CD16 monocytes. And the size of the DC6 subset over here seems to be a lot larger than UMAP plot suggests. And it turns out there are some interesting biological implications underlying these patterns. So both these cell types belong to the same developmental lineage originating from hematopoietic stem cells. And the monocytes are known to begin their lives as CD14 or classical monocytes. And then they go through a transitionary period before turning into a CD16 or non-classical monocytes. And Villani et al. Also have shown that different DC subsets, here shown as DC 6, 3, and 4, these can arise from different stages of this monocyte trajectory. Now, in this context, given this additional context, here's what our observations imply. So first off, this DC6 subset, this seems to have a lot of transcriptomic heterogeneity that was not previously appreciated. And if we look at this transition from CD14 monocytes to CD16 monocytes, this appears to be some type of specialization process where we go from this really diverse set of cells to a highly specific, highly concentrated cluster over here. And this is plausible given that CD16 monocytes are known to arise in response to an inflammation signal, where a small subset of CD14 monocytes go through a rapid transition to reach this cell state. And we have a similar observation going from CD14 or intermediate monocytes to the DC3 subset of dendritic cells, where we also start from this diverse pool of cells and we go into this highly concentrated area for DC3. So all these patterns were previously, I want to emphasize that these were difficult to see in a UMAP plot. And I want to make a quick note here that we validated some of these observations on 2 additional data sets where we were able to confirm the variability differences between CD14 and CD16 monocytes, and also between CD14 and DC3 subset. But we wanted to take a step further and try to quantify whether the visual density differences in our images truly reflect underlying patterns. So for this purpose, we tried to directly compare our local radius measure computed in the original space with those computed in the embedding. The points here are colored by different cell types, same way as before. And as you can see, the correlation coefficient for dense map is pretty high, around 0.7, whereas UMAP doesn't seem to convey density information at all, which is what we expected. And now the correlation in local radius is actually precisely what we asked the method to optimize. So in some sense, the fact that this correlation is high is not that surprising. So we tried to look at this from a slightly different angle as well, based on a more visual notion of density. And we call this neighborhood count, which is simply counting the number of data points within a certain radius around each point in the visualization. So this would be directly corresponding to what our eyes perceive when we look at these images. So now if we correlate this measure with the local density in the original data set, these are the plots that we get. Again, we see high correlation for dense map and low correlation for UMAP. So these results give us some reassurance that what we are seeing in these images are truly reflective of the underlying patterns. Now, we wanted to see if our methods could be used for finding disease-related insights. So this is a data set from Zilionis et al., including around 50,000 cells from 7 lung cancer patients, representing immune cells from both circulating blood and from tumor microenvironment. And this time, I'm showing you the results based on density and t-SNE. But for all these experiments that I'm showing you, we observed similar differences between these 2 pairs of methods that we compared. The colors represent different cell types as usual. And right off the bat, we can see that some of the clusters have clear differences between these 2 images and how much variability is conveyed. So for example, these plasma cells over here and neutrophils over here, they seem to be a lot more heterogeneous than previously seen in the t-SNE plot. But what we're really interested in looking at was to look at the changes in each of these individual cell types between blood and tumor. In other words, we wondered if we would see interesting variability changes as these immune cells migrate into the tumor environment and start interacting with the tumor cells. So here I'm showing you the same plots, but with different colors, blue representing blood and orange representing tumor. And once we start comparing specific clusters that belong to the same cell type between these 2 conditions, we start to see some interesting patterns. So here are some examples. So first of all, neutrophils seem to have comparable heterogeneity between tumor and blood, whereas in t-SNE, the tumor cluster seems to be much smaller. And now we know that this is mostly an artifact introduced by the limited number of data points in this cluster. And for T cells and B cells, we observe that as these cells go from blood to tumor, their transcriptomic diversity really explodes, with the immune cells in blood spanning only a small fraction of the range of states that these cells can take. And now, what is actually going on here biologically, we're not too sure yet, but I want to emphasize that we can draw these interesting observations about variability in ways that were not possible before. And even though we can see the same size differences in t-SNE plots, as I explained, this mostly reflects differences in the number of data points belonging to each clusters, not and not reflecting their heterogeneity. And just to give you a flavor of new kinds of analyses that could be motivated here, we devised a differential variability test that picks up genes that have significant differences in variance in gene expression between these two groups, which might be explaining this pattern. And here are some top hits that we get that explain the differences in T cells on the top and in B cells on the bottom. A particularly interesting example here is RPS27, which encodes MPS1 protein, which modulates activity of tumor suppressor P53. So for this gene, we observed a significant difference in variance in gene expression, but not in mean expression. So this is something that we would not have found using conventional differential expression analysis. So we believe that this really opens up a new direction in terms of looking into the biological processes that underlie these transcriptomic variability changes. The last case study that I want to go over with you is for a data set of C. elegans development. This includes 86,000 cells from all developmental stages of this worm. In the two figures on the right from DenseMap and UMAP, where the colors indicate developmental time points, you can see that the embryo starts from the central region and branches out into different lineages as it develops. And the plot on the left is a DenseMap where we colored things by cell type. And here we wanted to ask whether our methods could reveal temporal dynamics in transcriptomic variability. So to do that, we first looked at how the local density measure changes over time in the original data set for different lineages. And interestingly, we found two distinct subgroups with different dynamic patterns corresponding to different developmental models. So these three lineages over here, shown with solid lines, are thought to follow a clonal developmental pattern that mostly sticks to a hierarchical structure where one cell divides into a few daughter cells and each of those dividing into more cells. And we repeat this process, and we expect the overall diversity of this pool of cells to grow over time. And this is indeed what we see in our local radius plot. And on the other hand, these three lineages over here, which corresponds to the semi-clonal development, this intuitively means that there is more horizontal mixing in the hierarchy, for example, by cells moving around more freely in the tissue. So this population is better mixed, and therefore we expect there to be less of an increase in heterogeneity over time. And that's what we observe here as well. Now, the interesting part here is if we plot this data set using DenseMap and UMAP, these temporal patterns are more accurately captured by DenseMap, where we can see the same distinction between these two subgroups. But in UMAP, this distinction seems to be mostly lost. So here are some visual examples of this pattern, this temporal pattern. For hypodermis cell type, which is semiclonal, DenseMap shows a relatively constant variability over the trajectory, whereas in UMAP, it appears to decrease in variability. And now we know that this is because there aren't enough cells sampled from these later stages. Next, for the amphid cell type, which is clonal, DenseMap communicates that there's this expansion of diversity over time, whereas this pattern isn't as clear in UMAP. So I hope I was able to convince you that there are many interesting biological insights that our methods make visually accessible to researchers. And here I'm showing you the runtime of our methods compared to the original methods to show that the overhead is not too large here. Solid lines are showing our methods and dashed lines are the original methods. As you can see, the runtime overhead is relatively small across different data set sizes shown on the x-axis. And the overhead is around 30% for large data sets with hundreds of thousands of cells. And we think this is a reasonable overhead considering the value of additional information that we're adding to these images. I also want to point out that we tried applying these methods to other types of data sets outside of single-cell genomics. On the MNIST data set of handwritten digit images, we actually found this really interesting example where the cluster corresponding to ones become a lot smaller compared to in dense map compared to other clusters. And this makes sense given the limited degrees of freedom with which one can write the number one compared to other digits. And we also applied our methods to genotype profiles from the UK Biobank dataset. And here we saw that our visualizations better convey the rich genetic diversity of non-white populations in the dataset, whereas if you look at the UMAP figure, most of the visual space is given to the white population in the center around here, given that this population takes up the majority of the dataset. So these suggest that our methods could be more broadly useful for other domains beyond single-cell genomics.
Something particularly striking here and in some of the previous single-cell data sets is the way that some of the outlying clusters get squashed into these kind of one-dimensional shapes in the other methods and not in yours. Do you have any intuition for why why t-SNE and UMAP tend to do that? Like and I think people overinterpret those. They tend to see these things in single cell plots and view them as these very well-defined trajectories. Whereas in your plots, you show that they're actually just these big diffuse clusters.
Yeah, so we think this is mostly because there are not enough cells in those regions. The reason why they appear as linear trajectories, I'm not too sure, but all we know is that they're not these little strings in space.
Yeah.
It's actually much more diverse than we would expect.
No, for sure.
That's a surprising
people
part.
really tend to overinterpret those little strings, though. Yeah, Leland, I wonder if you.
Yeah, I can comment on the stringiness. They are more diffuse in the high dimensional space, but essentially what's happening is that there's really one primary direction of variance. If you think in PCA terms, it's almost like there's one major principal component, and then there's variability around that, but it's picking up that one thing. It might not be a nice linear thing. It might be curved in the space, but it's there, and so that's what these methods are keying on. Is given the limited data that you have, as Hoon has pointed out, it's doing the best it can. It's not trying to preserve the density. It's trying to presume that density is just uniform over the whole data set. And so it's preserving the primary variance structure, which is relatively speaking one-dimensional.
That makes sense. If in the high dimensional space, the first principal component captures substantially more variance than the second, which will generically be the case, I guess. So, yeah.
Yeah.
Okay, great. So I'm going to move on to the second part. Since we're running short on time, I'll try to be brief here. So in this part, I want to briefly describe GeoSketch, which is a method that we developed for sketching massive single-cell data sets for the purpose of quick exploratory data analysis. And this work was featured on the cover of Cell Systems last year. So the motivation of this work is this figure, which I'm sure many of you have seen, which shows the remarkable exponential growth of single-cell data sets over the past decade. And now it is fairly common to see data sets with hundreds of thousands or millions of cells. And of course, there are ambitious data collection efforts like the Human Cell Atlas, which aims to collect 10 billion cells across diverse tissues. So the scale of these data sets is expected to continue to grow. And the problem with this trend is that computational methods for analyzing these data have been struggling to keep up with the pace of data generation. So most methods are designed for relatively small scale data, including thousands or tens of thousands of cells, and they have difficulty scaling to large data sets. And this problem is made worse by growing needs to combine and integrate multiple data sets across multiple labs to gain a broader understanding of the given biological system. And for these massive data sets with millions or tens of millions of cells, even quick exploratory analysis can be quite challenging. So a common approach to tackling this issue is to take a small random subset of the data set for exploratory analysis, and then to follow up with a more targeted analysis on the full data. This is an example from the 10x Genomics website, where they provide a 20,000 cell subsample along with their large 1.3 million mouse brain cell dataset. for researchers to gain quick insights into this data set. And here's another example from a paper that introduced this clustering algorithm, which first works with a random subsample of 5,000 cells, and then later generalizes the clustering structure to the full data set. Similarly, for computational efficiency reasons. But an important concern for these approaches is that the process of randomly downsampling a data set is prone to completely excluding certain subpopulations of cells by chance. And rare cell types especially are unlikely to be properly represented in the subsample because there are so few of them to choose from. So an alternative way we could do this would be to use some type of supervised sampling where we explicitly specify which cell types to upweight and which other cell types to downweight in terms of their representation. This paper here is an example where the authors have tried to manually balance the cell type ratios in the data set for their analysis. But this could work well if we know all of the cell types in advance and know exactly what we're trying to do. But what should we do when we don't really know what we're expecting in a data set, which is true in most settings? Can we have a more unbiased sampling strategy? So to this end, we developed a method called geometric sketching, or GeoSketch in short, and here is how it works. So as we saw in the previous part, the single cell landscapes typically include both dense and sparse regions, as well as complex structures. And if we simply uniformly downsample the data set, then we're likely to miss rare cell types or fine-grained structures that may be biologically important. Instead, in our method, we first approximate the geometric shape of the data manifold by covering it with simple geometric objects, in this case, boxes or hypercubes in higher dimensions. And then we take samples uniformly across these boxes instead of sampling the points directly, which results in a more balanced sampling of data points throughout space. In other words, we sample according to transcriptomic diversity, not density. And this is the sense in which this method is density aware. So in the next few slides, I want to walk you through an example run of this algorithm to give you a better sense of how this works. So let's take a data set in 2 dimensions. And as I said, we want to cover these data points with equal size boxes to approximate their shape. So to do this, we first pick a direction. In practice, we're going to be using principal directions. So along this line, we're going to lay down equal width strips, like so. And note that after each strip, we skip over to the closest point, which can be viewed as a type of approximate greedy algorithm for covering these data points. So we continue doing that in this one direction. And after that, we choose another direction and repeat the process. And all these directions will be orthogonal to all of the previous directions that we used, just to make sure that these are we end up with boxes at the end. So once we're done and with this process in all dimensions, we get a covering of the data set with these boxes. And once we have that, then we can take even number of samples from each of the boxes, and that gives us a representation of the data set that gives us a more balanced view of the data. Okay, so to quantify, yeah.
Oh, sorry, you maybe mentioned, but how do you choose the first direction along which you draw the partitioning?
Yeah, so we run PCA and then use the PCs.
Great. Yeah, I think so. If you're using the principal directions, do you also use the eigenvalues to choose anything about the scale there, or not really? Do you have just a fixed R for all of the directions?
We did use the fixed R, but yeah, using a different R for each direction is also a good idea, but we haven't really tested this.
Thanks.
Okay, so here is how we quantify the quality of our sketches. We use this notion called Hausdorff distance. Let me skip over the detail, but here's an intuition for this part. So so overall, so we want to measure distance between 2 sets, right? We have the original data set and we have the sketch points. And we're going to measure it by the maximum distance that we ever have to travel to get to one set from the other. So this is an illustrative example over here. So let's say we have a set of houses and we want to place hospitals in the same region. And intuitively, a good representation, a good placement of the hospitals would be one in which from all of the houses in this region, the distance to the closest hospital is minimized. So this example placement is not a good one because the house over here has to travel quite a long distance to get to the hospital. But this alternative arrangement would be much better because this maximum distance is reduced overall. So this is the intuition behind this quality metric. And in our case, the houses are the original data points, and the hospitals are the points that we include in our subset. So we evaluated this metric on these 4 benchmark data sets and we compared our approach to the baseline uniform sampling approach, as well as 2 other baseline approaches that were proposed in the literature, which include k-means plus and SRS. And across the board, we can see that the Hausdorff distance for our method, shown in blue here, is a lot lower than all of the other methods in all cases. So this means that the sketch that we get is a more balanced representation of the data. And this is what an example sketch looks like on this data set of 500 cells of mouse brain cells. And this is a 5% subsample of this data set. You can see that GeoSketch leads to a more even representation of different cell types in this data set. And notably, there are some cell types that are significantly better represented in GeoSketch, including endothelial tip and microglia. And these 2 clusters can hardly be seen in the uniform sampling case. And this is because they are very rare. So if you just randomly sample, it's very easy to miss these points. And if we actually want, go ahead and count the number of cells in the sketch belonging to the rarest cell type in the data set, then this is what we get. So we indeed capture more cells from the rare cell type compared to the baseline approaches. And interestingly, we found that this ability to enhance representation of rare cell states is actually, this can actually help with biological discovery. So this is an example where we tried sketching this cord blood data set. And when we did a clustering analysis on our sketches, we were able to find this interesting subtype of macrophages that corresponds to inflammation response. And we had some validations here. The cool fact here is that if we were to do a clustering analysis in the original data set, then it's actually extremely difficult to cache this cluster because this is a really rare cluster that's embedded deep within this larger cluster of macrophages, which also include these orange points. So in this case, somehow by balancing the cell type ratios in an unsupervised way, we were able to enhance the biological signal in this data. GeoSketch is very efficient. Our runtime is quite close to uniform sampling, which is trivially fast. And even for very large data sets, for example, including tens of millions of cells, the runtime is around 1 hour, which is reasonable. And one other thing that I want to point out here is that we can use our sketches to accelerate other downstream analysis tasks. So this is an example of data integration where the goal is to combine multiple data sets that are independently collected by different labs. Here I'm showing you 2 data sets here, orange and blue. Harmony and Scanorama are 2 methods that were proposed recently for this task. And you can see that they output a combined data set where some of the batch effects for these data sets are removed. So what we can do here with GeoSketch is that we can actually run these algorithms in a small sketch of this data instead. And then once we have that transformation, we can apply that to the full data set to get a fully combined data. And by using this approach, we were able to get down this runtime from 2 hours to 8 minutes without sacrificing integration quality. And lastly, I want to give a quick plug for this follow-up paper from Ben and Bonnie, both of whom were involved in this GeoSketch work, where they came up with a new algorithm for sketching that achieves even better Hausdorff distance, along with some nice theoretical results. So if you're interested in this direction, please check out this paper as well. The software for both projects that I described today are freely available for you to try out. And in fact, DenseMap is now available through the Python UMAP package, thanks to Leland, our next speaker, who maintains this repository. And with that, I want to thank all of my co-authors on these projects. I thank these funding agencies, and I thank you for your attention. I'm happy to take more questions now.
Wonderful. Thank you so much, Hoon. I think we have time for at least 5 minutes of questions. So opening the floor for anybody to chime in.
Hoon, could you throw those links on the previous slide into the chat when you have a minute? Or just leave the slide up, either one, yeah.
Sure, that's easier.
Maybe I can start actually, unfortunately, by asking you to move one slide earlier, or wherever were the results of the sketching and then alignment.
Yep.
Right. I don't know how to interpret this, but it just occurred to me as I was looking at it that with the strategy of first doing sketching and then the two different aligning strategies, the results on the entire data set look a lot more similar between the two approaches compared to when they're applied to the entire data sets. Or maybe I'm just over interpreting Tisney's likely.
Yeah, I'm not sure if I see the same observation here. I would say maybe it's hard to see on the screen, but I think this result is still closer to the original output than it is to the harmony output, I think. For instance, if you focus on this area, this part is a little more orange than what you would see over here. I'm not sure that if it's making the two methods more similar. Yeah.
Yeah, I'm not exactly sure how I would even quantify how similar the outputs are. But yeah, I was just going by some visual kind of resemblance.
Oh, did you mean similar between the two data sets instead of between the two methods, like no, between blue and orange?
No, I meant between the two outputs of the two methods. Mm-hmm. I was just looking at like the little like orange feathers in the bottom right. Like now they're like both there, or the like little windmill like looking thing in the top.
Yeah. Yeah, I mean, I think like because both of these visualizations are based on the same set of data points, I think if you wanted, you could define some sort of measure of how similar the placements are, right?
Sure.
Based on the neighborhood of each point.
Yeah, I'm not sure what's like a biologically useful way to think about that though.
Yeah.
Okay.
I have a question that's more about the dense map side of things.
Sure.
I'm James Weber. I'm a computational scientist. So one aspect that could make things seem less dense is like just the sampling depth of the cells, because they'll have more noise and maybe seem farther apart from each other. Do you think that can like affect the result of the density embedding? Because in some cases you might want them to appear equally clustered when it's just a matter of library size or something like that. It's more of a technical artifact. I don't know if that's something that you looked at or.
So something like that would affect the density of the entire data set, right? And so if you apply these density preserving methods, it's not really clear whether it would actually change the visualization by much, because it's mostly focusing on relative density differences between like different regions in the data set. So if you reduce the density at the global level, it might not have much of an impact on that.
Yeah, I was thinking if there was actually a difference within your data set between different cell types or something, with like some sort of sampling difference between cell types in a data set. I don't know. I was just thinking about like other reasons that your data can appear tighter or more variable beyond kind of the biological heterogeneity.
I see. Well, so the idea is to remove this confounding factor, I think, right? So if you have one cluster that you're trying to plot in the image, and somehow we didn't sample enough from that cluster, if we just use UMAP or Tisney, that cluster would appear much smaller, and just because of sampling density, sampling rate, right? Because there are not many cells there. But if we incorporate the density term, then it's more likely. I can't guarantee. We would need to do like a more comprehensive test here, but it's more likely that the overall shape, the size of the cluster would remain more similar, even with the sampling rate effect.
Yeah, we'll talk later, so we could talk about this more.
Sure. Yeah, I think we have a meeting later this week.
Yeah.
Okay.
I had a question. It's a great talk. So one of the common questions that comes up with UMAP and Tisney is that they don't always preserve global distance very well. Do you think incorporating density information can address that to some extent, or is that not something you can measure?
Well, in my opinion, they. Wait, did you mean global distances?
Yeah, like global distances within the embedding.
Okay. Yeah, they're good for local distance, but maybe not for global.
I guess I'm wondering, does density do a better job of representing global distance in the original dimensionality?
That would be nice, but I can't say that we expect that to be the case because we haven't, in our design of the method, we didn't do anything that's specifically addressing this issue of global organization of clusters. And because we're mostly relying on the same manifold approximation that t-SNE and UMAP is, are using, I think in that respect, I don't expect to see an improvement in that regard, unfortunately.
I think there's a real tension there. When you're going from high to low dimensions, you sort of have to compromise on the larger scale directions. Like you can try to preserve local structures, but if you try to preserve global ones, you're kind of back to PCA, and then you have problems, right? Like this is kind of what motivated t-SNE and UMAP math in the first place, these issues.
Yeah, so you might be interested in looking at this slide over here. So we're trying to vary the weight term that we discussed earlier. So yeah, this is exactly the issue that you're alluding to here, where if we try to preserve density too strongly, then we end up losing some of the structure, right? And this is precisely because there's like not enough space in the image to place things. We want these parts to be really, really sparse, but once we try to do that, it ends up like merging into nearby clusters, which is not ideal, but there's just not enough space. And this is slightly better if you plot this in 3D, but at some point we're going to run into the same issue. So I think like really the conclusion here is that we can try to infuse density information, but not to the full extent, but maybe that's okay. Like maybe we just need some indication that there are real density differences here to the extent that we can glean from these images.
Thank you so much for inviting me to speak here. So I want to preface this talk with a caveat, which is that I'm a mathematician and I do a little work in machine learning and data science as well, but I don't know much biology at all, and certainly not specific details of working with single cell data sets and so on. I have learned some of the basic shapes and kinds of data that show up in single cell just because some of the packages that I've worked on, like UMAP, have been used in those fields. And from that, I think there's some really useful crossover that can come from natural language processing solutions, NLP. I'll often refer to it as NLP for natural language processing. And I think those techniques can be really useful for single cell. It's not immediately obvious, but I think they can be. And I really hope that this talk can just be the start of a conversation that can spur some ideas about how we can bring some of these tools across from a field that I'm currently working in, sort of machine learning on natural language processing, to single cell biology. So first of all, let's just talk about motivation. Why am I talking about this at all? And at least part of it is that natural language processing, NLP, has seen some amazing advances recently. And these have, a lot of them, been driven by fancy new neural network and deep learning based techniques, but they can produce really stunning results. So here's a typical tweet about BERT, which is a huge deep learning based language model from Google. And they're saying it's transformed the landscape, it's seismic impact. And this isn't hyperbole either. BERT and related predecessors like Elmo really have completely changed the natural language processing landscape. And it's certainly a field that's big on hype is AI, but a question you want to ask yourself to sort of clear away some of those concerns that it's just an overhyped thing is it used in production? Is it actually used in practice? And the answer is that Google actually has shifted over to using BERT in language modeling in all their search queries. So it really is transformational in a way that matters to Google, because they don't mess around with their search technology unless it's demonstrably improving their bottom line, demonstrably improving the search. So it really is a huge impact, and it really does matter. Another example is this tweet talking about natural language processing. A lot of traditional techniques had topic modeling approaches that if you squinted at it in the right way, they could be interpreted as meaningful. But that usually required all manner of careful pre-processing. There was lots of language-specific stuff. So if you wanted to work in different languages, it was very hard. But more recent techniques like the universal sentence encoder, again from Google, that was talked about in this tweet, produce results that just work. There's no fine-tuning through elaborate pre-processing, no sifting the tea leaves of the topics to try and produce some sort of sensible interpretation. You just get things that make sense straight away, straight from text. So from the perspective of natural language processing, there's been huge advances in the last few years that have really changed the landscape entirely. But these are all fancy deep learning based techniques. BERT is a giant, complicated, transformer-based neural network model with literally hundreds of millions of parameters. Universal sentence encoder is a giant pre-trained model that you download as a black box. It's got hundreds of millions of parameters. And if you just download the basic model weights and all, it's a 1 gigabyte sized model that you have to work with. So if you don't have neural network expertise and your data isn't text, these models don't really help you or don't seem to help you. But a question we can ask is, how far can we get by reverse engineering the underlying techniques, the things that are really making these advances work, and just try and use those simple core ideas and build something simpler that hopefully gets us a lot of the same way to the right end result. And what I want to emphasize here is if we take the simpler approach, I feel like it's much more generalizable to many different types of data. And I believe that will hopefully include single cell data. So to get started, we need to work with embeddings, because this is the core idea that's really revolutionized a lot of the NLP space.
Hey, Leland.
Yes.
This is Brian. I just want to, well, this is not a question, frankly, to be honest. I just want to interject on the importance of the last thing you were just saying about the applicability to many different applications. Mm-hmm. I think that's like absolutely critical as a field, that we focus on things that can be sort of adapted to many different tasks. Because if you just have one huge model, then it's not very useful for very many people. There's one thing, one entity that owns that, but it doesn't really advance an entire field. So yeah, I'm like, I just find the point you made super important. So I just wanted to sort of bump that.
Excellent, thank you. All right, so the core idea here is embeddings. And the question we need to ask is, what are these magical embeddings that have powered of this NLP work? These word embeddings, sentence embeddings, contextual word embeddings, and embedding in the context of these NLP methods. What we actually mean at the end of the day is a combination of a mathematical representation of the data and a way to measure distance between those representations. Now, when I say mathematical representation, because it's the easiest possible thing to work with, we almost always mean vectors. And that's what's going on there. What we care about is the way to measure distance between them as well. So a lot of the focus and talk, if you look at the literature around this, on say word algorithms like word2vec, which you may have heard of, that's again from Google. People spend most of their 100 to 300 dimensional dense vector representations of words, and they elide the fact that what really makes them useful is that we have a sensible measure of distance between them, and that measure of distance is what really gives meaningful information about these things. And in practice, distance is critical, and we're going to see that later on. So now that we've got the basic idea of embeddings, let's move on and start looking at some specific cases. Now, I'm going to spend a lot of this talk talking about NLP-based things. So I'm going to talk about documents and words and sentences and so on. Please bear with me. I will try to interject whenever I can how I think it ends up applying to single cell. But for at least the first little bit, it's going to look like it's really just text, but it'll hopefully start to become clearer later how this is going to work out. So I'm going to start off with document embeddings, because these are actually coming from decidedly old school classical natural language processing techniques going back 30 years or more. And it'll give us a pretty easy introduction to the whole thing and make clear what sort of data we're really dealing with, because we're going to transform the text into something else, and then that's pretty much all we're going to work with from there on out. So how do you represent a document mathematically? How do you turn a pile of words into vectors? And the classical approach for this, which is still around largely because until very recently there were very few things that were actually better, is surprisingly simplistic. And it's what's known as the bag of words model, or bag of words approach to doing this. And it makes a lot of pretty nasty assumptions about documents. First of all, it throws away word order. Now, that's a big assumption because clearly the order of words in a document really matters. But it turns out that you can do a lot without that word order. You can still manage to do a lot of things, and they still work even if you ignore it. And it just is the thing that makes it simpler enough to work. So let's see how this whole bag of words approach to turning things into a vector works. So here are a couple of simple documents. In this case, it's just auto-generated filler text so that it's all pleasantly meaningless. We want to convert this into vectors. So the first thing we're going to do is strip away all the order, the punctuation, everything, and consider each document a bag of words. And they're just random words sorted alphabetically. Okay, that's a start. That's still not vectors, though. But what we're going to do is just count how often each word appears in each document over the whole set of words that we have between all our documents. So we can make up a table. So here I'm going to label the columns by the word, and then for each document, I'm going to generate a vector, which is going to count how often that word appeared in that document. So nothing fancy here. None of the words appear in any of these documents more than once, so these end up looking like binary vectors, but I have a vector for each document, which is just the list of or count of how many times each of these words appeared in the documents. It really is that simple, but it's surprisingly effective, and it certainly becomes more effective as you get to longer documents. So here's a giant wall of text. Don't worry, you don't have to read it. This is, again, just auto-generated filler text. It's 5 paragraphs. I'm going to consider each paragraph a document. And we're going to go through the same, to make a big vocabulary list of all the times each word gets used in each document. And the result of that, well, now I'm going to visualize the matrix I get out as a heat map. And what we're getting is a large sparse matrix of counts of how often words are observed. And at this point, I hope you're looking at these results and starting to think that perhaps this looks a little like some of the kinds of data that you see in single cell work. Large sparse matrices of counts of things. Each row here represents a document, but it could equally well be a cell. And each column is, in this case, a word, but it could be just something that's occurring in the given thing. Maybe that's gene expressions or so on. So in this case, some of the words occur quite a lot and show up in most of the documents. Other words are very, very sparse. The counts are pretty widely varying. This is, I think, hopefully something that looks like single cell style data in many ways. And this is kind of my point, is that while we start with this notion of text and documents and words, we're really just dealing with these large sparse matrices of counts. And the techniques applied to this should be very generalizable. Okay, let's scale up and have bigger data sets so we have something to play with. And this is a data set I'm going to keep using in examples from here on out. And so this is a famous toy data set from natural language processing called the 20 news groups data set. It's a set of posts from old-fashioned NMTP news groups. Hopefully some people are old enough to remember those. In total, there are around 18,000 posts, and hopefully they're collected from 20 different news groups. And the classical NLP task being, you want to predict which news group a post was from based solely on the text of the post. Now, in practice, we'll use it just as a marker for how well we imagine the data ought to divide up, roughly speaking. And if we get things right, we should manage to be able to split out the different news groups very easily. But there's also a hierarchical structure here if you want to look at it. If you look at the list of groups there are actually sort of like major types and then subtypes within that. So the major types, there's a whole bunch of groups talking about computer-based stuff. There's a bunch of groups that are all talking about different kinds of politics. There's groups of news groups that are talking about religion in general, and so on. So you've got like type relations. Again, I think this is the sort of thing that tends to crop up in other fields as well. And if we look at the results of the first 1,000 posts from that, I had to log transform the counts to make this show up visibly for you, which I gather is a thing that often happens. But what we're getting is a large sparse matrix of counts. And I hope you're looking at this picture and saying, well, this looks like a lot of the pictures that I tend to see in single cell data sets. They tend to look like this. They're sparse. We're often log transforming things. They're really just counts under the hood. And that's what's going on. So just large sparse matrices of counts. I want to emphasize this looks like a lot of other kinds of data. I think there's a lot of places we could actually apply these techniques to. Now,
Leland,
yes, please.
I don't know if this is a very well-formed thought, but I just want to challenge that a little bit. In that I feel like there are underlying continuous latent structures that are very different between these settings, even if the manifestation ultimately as counts is kind of converges. The kind of the underlying latent structures that lead to the, like, eventually you're sampling things and so you count, but there are underlying structures behind single cell that are vastly different from those behind language.
I understand, and I certainly agree. My hope is that the ideas behind these techniques are still applicable because they rely a lot less on the underlying latent structures per se than just generally being able to process it in certain sorts of ways.
I see.
So I would certainly be interested to discuss this. Bear with me for now.
Oh, for sure.
And we'll hopefully come back to this because this is exactly the sort of point where I don't have the expertise to understand the details of the data you have and how applicable these techniques will be. But
sure. Just to offer a little intuition, I feel that a lot of people think of the single cell as that there are these kind of underlying continuous quantities reflecting the transcriptomic state of a cell, and that's in some sense that's what you're actually trying to get at.
Yeah, but you end up with measurements based only on counts. Is that fair?
Indeed, yeah, that are very much downsampled, and
yeah.
And in some sense, even the truth is discretized because there's a discrete number of mRNA molecules of each species, but that's actually not the discreteness that you deal with. You deal with the discreteness from the severe downsampling that you get from the limited capture in the technology.
Okay, yeah, that's fair. So I would argue perhaps one could make similar sorts of arguments about language. So one might suggest that in language, there's sort of a continuous structure. Words have shades of meaning in a continuous scale, a very complex continuous scale. but all we get with text is a discrete set of measurements of that. There's maybe a deeper things going on, but all we have is the text document, which is some discrete realization, observation of some underlying meaning. So it's not an exact match here, but I feel like there's I think what I'm hoping for is analogies rather than direct matchings.
Sure. Okay, great.
Okay, so that was our mathematical representation. I said distance really mattered. So how do we measure distance? Well, if we look at these documents as distributions of words, we probably want a distance between distributions. And as distributions, they're going to be multinomial distributions. And the way to measure distance between multinomial distributions that makes a lot of sense is a distance called Hellinger distance, which derives from Fisher information distance on manifolds of multinomial distributions. Hellinger distance essentially is a good approximation to that. And in NLP applications, you'll find that people will almost always use a different distance. They will use something called cosine distance, which I've shown just below. And cosine distance, if you stare at it, looks a whole lot like Hellinger distance. It's dropping off some square roots, and it's using L2 norms instead of L1 norms, but it's very, very similar. So actually, it's very closely correlated with Hellinger distance. If you line up a whole bunch of data and measure distances between them, they essentially are almost the same, except for a little variation here and there. The main advantage that cosine distance has, and it's why it'll crop up later and we will be using it a little later, is that Hellinger distance is only defined for positive quantities. So when we're dealing with counts, all counts are positive, everything is fine. But if you have negative values in your data, Hellinger distance is going to break down. Cosine distance will be perfectly valid there and everything will work just fine. So that's our notion of distance. And let's use that distance in UMAP to take the 20 newsgroups data set and embed it into 2 dimensions so we can see how this mathematical representation looks with this notion of distance. So this is our sort of straightforward document embedding. And you can see it's done something somewhat reasonable. It's separated out some of the more computational news groups from some of the more politically and religion-oriented news groups, but it's still a bit of a big blurry mess. I should note, if you check the bottom there, UMAP with metric equals Hellinger, that's actually natively available in the current release of UMAP. You can just specify Hellinger distance and it'll understand and use that.
Leland, if I may interrupt you for a second, there is a discussion in the chat. If you can go back to your previous slide, it's just to clarify the notation where you have, for example, xi, comma, yi.
Oh, sorry, there should be no comma there. Yeah, no, sorry, that's a multiplication. I don't know how a comma slipped in.
Like like a multiplication. So
yeah, sorry. Just multiplication of the values. So, I mean, it's called cosine distance because this is the dot product of the vectors x and y divided by their L2 norms. So this term here is giving essentially the cosine of the angle between the vectors, and then to turn it into a distance so that vectors that are the same have distance 0, we're just doing 1 minus that value. Hellinger distance is doing essentially a similar trick, but it's the more statistically valid approach if you're working with multinomial distributions. Sorry for the confusion in the notation there. I apparently prepared my slides poorly.
Thanks for clarifying.
Okay, so that was just the raw representation with Hellinger distance, but every domain has its domain-specific transformations that they apply to the data, dealing with different data types, because different data types can get distorted in different ways. Now, that might be dealing with batch effects in single cell data, or highly skewed counts, or all kinds of things. And so my point here is that There's different kinds of transforms you're going to need to do on your data. And this part, I believe, is entirely domain specific. And you're going to have to work with what makes sense for your domain. You're going to have to do something that's going to operate on the data and the raw counts to sort of align it with your expectations. And in natural language processing, the popular trick is called TF-IDF, which stands for term frequency inverse document frequency. So the term frequency is just the count of how often the term appears. And inverse document frequency is essentially we're looking at how many different documents the term appears in, how frequent it is across the range of documents that we have. So the thinking here is that terms like the and all those common words, they're going to appear in all the documents. So they're actually not very interesting. They don't help differentiate our data very much. So we want to downweight them. So they're very document frequent. So we're going to essentially divide by that document frequency, hence inverse document frequency. Whereas less common words are going to show up in just a few documents. And more importantly, you really want to give specific weight to words that only show up in specific subsets of documents. This is the natural language processing mentality here. I don't want to get into the details of TF-IDF. There's some fine points, some logs that show up for, it's hard to explain what reasons. The key here that I want to get across is, yes, there's a slot right here where the domain-specific transforms that matter to your data need to get inserted. And really, it doesn't matter too much what those transforms are, as long as you still have a useful notion of distance between the transformed results. And because TF-IDF is just essentially reweighting columns, we're still working with essentially multinomial distributions. So we can still use the same Hellinger distance. And that's often the case with a lot of these transforms. You're just integrating or rescaling or reworking the data, and the same underlying distance function between the representations is still the valid approach. So if we take 20 news groups and apply this TF-IDF transform to it, what we get out under UMAP is this. So still sticking with metric Hellinger, now we've separated out the data quite a bit better. It's done some very useful things. The major types are much more cleanly separated. We've got all the computational news groups in sort of reds and oranges down in the bottom left, and then the more religious and political ones in the sort of upper right. It's separated out the sport-related ones in that sort of reddish, brownish, beige color. It's still kind of a bit blurry and messy, but it's certainly improved the data over the raw matrix of counts that we started with. But I said at the beginning that distance matters. And Hellinger distance or cosine distance assumes that we're working with multinomials. And that in turn assumes that we have independent columns, that each of these columns is independent from each of the others. But are my columns really independent is the question. And the answer is no. Some words are quite similar to others, while other pairs of words are entirely orthogonal and distinct and have no relation to each other. And the same, I would imagine, and this is where I'm hoping for some feedback, would apply to the column spaces for single cell data. I would imagine some genes are more similar to others, and there's sort of relationships among them. And as long as that column space matters in this way, as long as it's not really entirely independent columns that can be thought of as entirely orthogonal to each other, I think all of this still starts to go through. So if our columns aren't independent, what are we looking at? Well, here's two short documents that are very carefully chosen for this specific demonstration. But we don't need to go through the trouble of encoding them in a bag of words model to see where we're going to run into difficulty. These two sentences have absolutely no words in common. So when we're looking at them in the bag of words model, there's going to be absolutely no overlap, and their Hellinger distance is going to be maximal. Their cosine distance is going to view them as completely independent of each other and orthogonal. And essentially, these are going to be about as far apart as two sentences can be in our data. But on the other hand, if you look at the sentences, you can quite quickly see that they are, in fact, very, very similar to each other. And that's because their columns aren't independent. Red and scarlet are similar words. We could replace one with the other without much loss. They're pretty close to each other in terms of meaning. Rose and flower are pretty similar in terms of their meaning. And so this lack of independence in the columns really, really matters. And what we're doing by treating the columns as distinct categories is losing all of that information. And that goes for words and sentences, or however you want to do this. Treating our words or the columns of our large sparse matrix of counts as independent is problematic. So let's move on to word embeddings, because this is the way we're going to find the structure of the column space, so that rather than treating it as independent columns, we can improve our understanding of that column space and start to tweak our notion of distance to be more informative. Now, word embeddings sort of really picked up in popularity in the NLP space when Google came out with an algorithm called word2vec, which is a neural net-based algorithm for generating dense vectors associated with words. But what I'm going to go through here is an alternative explanation of word vectors, which actually runs pretty similar to the underlying theory of what's going on inside that neural network of Google's, that I think is a lot simpler than you'd expect, and actually pretty much as effective. So the core idea behind word embeddings really is summed up by this quote from the linguist John Firth, which is essentially the claim that a word is ultimately defined not by some magical innate meaning of the word, but by the kinds of words it gets put together with to make sentences. Essentially, words are defined by their usage. So another way of thinking about that is, if you were to encounter a new word that you didn't really know and you wanted to get some sense of it without having a formal dictionary definition to hand, you might ask someone, oh, can you use it in a sentence for me? Maybe, can you use it in a couple of sentences? And after enough sentences that use that word, you can infer by context what the word has to mean. So the goal is to define a word in terms of its context of use and simply collect up all its usages and all the associated contexts of those usages and hopefully discern meaning that way. So here's what's really going on in all these modern word embedding algorithms. They all work essentially this way. There's fine details. We won't worry about those because, as I've been trying to communicate, I think that the core ideas are very translatable to other domains. The specifics for text, well, that's another matter. So the goal here is that we have a target word, which I've got in the middle there, and then I've got some context around that word. You choose a radius for your window that you want to use to define context. Some algorithms might keep the before and after contexts separate. Others might just lump them all in the same pile. But really, what we're going to do is go through our entire set of documents and scan them and just see for each given target word, what are the contexts that show up around it? And I'm just going to collect those up. So a way to think about that is I'm going to represent a word as the document, and that's document with scare quotes in some sense, of all its contexts. So in other words, I'm just going to scan through my entire bunch of text. And whenever I see a word, I'm going to look at the words that occur around it, and I'm going to take those and add them to the document for that central target word. I'm just going to keep building up this document. It's not going to be a document that makes any sense. It's just going to be random smatterings of context around wherever that particular word occurs. But it's the information about how this word gets used. And we're about to dump all of this into a bag of words model anyway, where we're going to throw away everything like order. So it doesn't really matter that the document as such of all its usages doesn't make sense. we're just going to convert it as documents into vectors in the manner we already discussed. We're going to throw away all the order. We're going to throw away everything. We're just going to count how often other words occur around each given word. So another way to think about that is that the matrix that you're going to get as a result is the matrix of counts of how often pairs of words co-occur nearby to each other, where nearby is some specific radius window in the case of these word embeddings. But the key idea here is that of co-occurrence. We want to count up when two things co-occur with each other, and that you have this notion of nearby for that co-occurrence. So all we really need is co-occurrence as a notion and nearby as a notion, and we can do this. And then what we're going to get is a giant sparse matrix of counts. And there's nothing actually very specialized to words or text or language about any of that. We just need co-occur, nearby, count up how many times these co-occurrences happen. And then basic operations on the resulting matrix, followed by a singular value decomposition, are enough to produce good word vectors. Now, you'd think surely this isn't giving us word embeddings as good as word-to-vex neural network or deep learning-based techniques, right? Well, actually, this is 90% of what's going on inside that neural network. And if you choose your matrix operations carefully, where the ones that I've been using lately, you column normalize the matrix so that the columns sum to one probability of a word. So I'm essentially getting the probability of a word given a context. Then I'm going to row normalize that so the rows sum to one. Now I've got essentially the probability of a normalized context given a word. And we're in a position to essentially have multinomials. We could apply Hellinger distance, but we want dense vectors. So we're going to apply this singular value decomposition. But singular value decomposition is implicitly assuming a Euclidean metric over the rows. And we can cheat around that and do a quick transform. So you remember there were a couple of square roots in the Hellinger distance formula. We're basically going to apply those. So repeated square roots, or in practice, a single fourth root to make up for that square rooting in the Hellinger distance, to make Euclidean distance a good enough approximation to Hellinger on that processed data. And applying the singular value decomposition to that, you get dense word vectors out. And actually, they look and behave just as well as those produced by Google's word-to-vec algorithm.
This is really cool. Is this written up somewhere? Like the verbal description was kind of tantalizing.
So this particular version is not written up somewhere. There was a paper at the NeurIPS conference quite a while ago by Levy and Goldberg on word-to-vec as implicit matrix factorization, which outlines a slightly different version of this, but basically views word-to-vec as a matrix factorization algorithm, and what matrix are they factoring and so on. This is what's going on. So I will talk a bit more about this later, but yes, this is surprisingly simple, and it actually produces word vectors pretty much as good as word-to-vec. But as you notice, here's the code to do it in Python in just a few lines on the 20 News Groups data set. It took a minute and a half to run. And I'm really doing nothing other than counting up how often words co-occur with each other within a window radius of, as it turns out, 20. I've got some extra bits and pieces with harmonic kernels and whatnot. And my normalization is maybe a little different than what I described there because we've been playing around with this a little. I'm taking fourth roots, I'm doing a singular value decomposition, and I'm pulling out word vectors. So this is very simple. And as long as we have co-occurrence and nearby, we can do this on any sort of data you want. So my questions for you would be, what does co-occurrence and nearby mean for single cell data, and whatever the column or space, the alternative to the thing you want to embed. Because I noticed Hoon had the so transpose one way or the other, column or row stances. But the short answer is that this is word vectors, but in a very generalizable context. So how do we make use of them? Before we loop back to our document embeddings with the column space solution in hand that we have here in these word embeddings, just based on local cooccurrence, we need to take a little diversion because we have to work out how we're actually going to make use of them. And this brings us to a topic called optimal transport. So we want to measure distances between distributions. That was at the heart of what we wanted to do. We need to understand how to do these distances better. Well, how do we measure distances between distributions? There's Hellinger distance, which I already mentioned. There's Kullback-Leibler divergence. There's total variation. There's actually innumerable different ways of doing this. But all of these ones that I described here have the same problem, which is that they really want the distributions to have common support. In other words, they expect the distributions to all be sort of filling out the same set of variables over the same full space. And in terms of our distributions of words, as long as the two distributions have a lot of words in common, all those measures are going to be okay. But as we already saw, that's treating the columns, the different words, as completely independent. And those two sentences I gave that had no words in common, all of these definitions of distance are going to fail in that case because they're just treating them as independent. So the right measure of distance between distributions, if we care about these things, is something called Wasserstein-Kantorowicz distance, or sometimes just Wasserstein distance, sometimes just Kantorowicz distance. The core idea here is actually pretty simple, and there's a lot of deep underlying theory that actually makes it very nice. If you want to look up lots of fun math, I highly suggest you Google the term Kantorowicz monads, because they'll explain to you how all of this works in some fancy categorical setting. But what it boils down to is that we want to measure distances between distributions where the distributions themselves are defined over a metric space. Now, if that sounds a little abstract, it'll become clearer with just a couple of examples. So here's a simple example. Here's two distributions, blue and red, shown as histograms. And as you can see, the common support, the range of values that they share mass for is pretty small. The regions where they overlap, it's not that large. And so our sort of previous measures of distance aren't going to work very well for this data. Because they only care about the regions where both distributions are defined, and not at all for the regions where it's not. So how do we want to measure the distance between them? Well, one could ask how far we'd have to move one distribution to make it look like the other. So note that this is fundamentally making use of the fact that we have a notion of distance along the x-axis there. And that's what I mean by distributions over a metric space. We know about moving along the x-axis. So how far do we have to move things to get the blue distribution to look like the red distribution? This is actually, if you come from a computer science background instead of a statistics background, called Earth mover distance. And the way to see that is to take that red distribution and flip it upside down. So the way I want you to think about this is that the blue distribution is a big pile of dirt, and the red distribution is a big hole in the ground. And because they're both probability distributions, they both have total mass 1. So we know, a priori, that the blue pile is exactly as big as the red hole. So our goal is to move all the dirt from the blue pile and fill in the hole. And ideally, we want to do that with as little work as possible. So our goal is literally to move the Earth and fill in the hole, hence Earth mover distance.
A nice historical note is that Monge's formulation of that problem in terms of dirt and a hole goes all the way back to the 18th century.
Oh, very nice.
The Earth mover picture actually came before Kantorovich, figured out the linear relaxation. The convex relaxation a couple hundred years later.
Excellent. So you're way ahead of me on all of this. I'll keep going anyway. If we want to think about the amount of work it's going to take, well, work is force times distance. Our force here, we're going to just assume is effectively constant, so that, acceleration is effectively constant. So our force is just proportional to the amount of mass we have to move. So it's really just the mass times the amount of distance we have to do it to fill the hole. And we want to find the least work to fill the hole. And that's really all that's going on here. And so our measure of distance is going to be the amount of work to fill the hole with the least, given that we're trying to find the least amount of work. How do we find the least amount of work? We want to find the optimal transport plan, where a transport plan is just a scheme defining which mass gets moved where to fill the hole. And in practice, this can be solved by linear programming. So for one-dimensional distributions, that's actually really easy. For higher dimensions, it gets harder, but it's still a linear programming problem. You can still just push it through a computer and solve it. And notably, it still makes perfect sense in higher dimensions and other metric spaces. So there's plenty of useful theory on optimal transport. This is just a small primer. As Alex mentioned, there's a whole slew of theory going back hundreds of years on this. It's a deep rabbit hole to go down. But just précis over it, all we care about is the fact that there exist solutions to this optimal transport plan to measure a distance between these distributions for how much work it is to move from one to the other. So just looking at it as distributions in higher dimensions, where high means 2 for the purposes of visualization, but feel free to imagine it in 100 or 1,000 dimensions if you want. We want to measure the distance from the orange distribution to the blue one, and we can do that by solving the optimal transport plan and then just adding up the amount of work it takes to do that and tell us which data gets moved where. So just starting to fill in pieces of the plan, here's the optimal transport solution that got proposed. And so you can see, it's not always best to move a point to its nearest point, because that's going to leave some other stragglers behind that you're going to have to move further. But it's going to match things up. And you'll also notice it's sometimes splitting masses up. A single point might have multiple destination locations. That's okay. The total mass just has to be conserved, and that's all that's going on. We fill in a little more, and you can see how the plan is matching everything up nicely with a little bit of splitting here or there. We can fill in all the rest, and you can see the full plan. It's a little harder to make out because now we've got so many crossings, which is why I sort of built it up in a few different stages there. But this is the plan to move the orange points to the blue points in the least amount of total work. And you can also picture that as literally, here we have the green point moving from the blue distribution to the orange distribution. Note that we're moving to the average of the destinations in orange, which is why some of those green points end up in the middle of the circle there. A fixed blue point got moved to a couple of different orange points, and we're just realizing it as its average there. Now, computationally, this is expensive, but there do exist ways to speed it up. So Sinkhorn iterations and linear optimal transport are a couple of the keywords you want to search for if you're interested in these sorts of problems. So now that we've got a measure of distance, let's bring it back to this joint Word document embedding approach. So path is pretty clear. We model a document as a distribution of words. We can consider the space of word vectors and distances between them, and using Wasserstein distance, get a much better measure of distance between the documents. So again, I want to emphasize that while I keep saying documents and words, there's nothing inherently text-oriented in any of this. We had a large sparse matrix of counts, which we pre-processed with our TF-IDF, but whatever domain-specific pre-processing you want to use. And really, that large sparse matrix of counts is the observed data we got from some underlying thing. We threw away all the word order information, for example, in our bag of words model of the document. We also have this cooccurrence structure of the column space of that matrix, which we use to define a measure of similarity between those columns. And now we're just using Wasserstein distance as a much smarter way to measure distance between the rows. So all of this, I believe, goes through just as well for all sorts of general problems. And I believe a lot of the single cell data will be amenable to these sorts of approaches. That's my hope, and hopefully we can discuss that more later. So let's reconsider that problematic pair of sentences that previously we considered to be utterly distinct. So presuming we learn a good word embedding, and the techniques I've shown here will definitely suffice for this. They will produce this as the optimal transport plan for these things, providing we have a reasonable sized training corpus to learn word embeddings from. So it's going to match up the words like this. So this is going to go to that, red to scarlet, rose to flower, smells to scent, sweet to lovely. There's a bit of reordering there, doesn't really matter. The Wasserstein distance between these sentences will be small because each of those pairs is pretty close. Red is very close to scarlet in the word embedding space. Rose is pretty close to flower in the word embedding space, and so on. So that's nice in theory on contrived examples, but how does it work in practice? So I learned word vectors directly from the 20 newsgroups data with exactly the code I showed you, not anything complicated and not doing any fancy word-to-vec tricks, just counting up co-occurrences, applying some basic matrix operations, and getting some vectors out. And then ran that through UMAP using Wasserstein distance. And this is what I get out for the embedding of the 20 newsgroups data. So this is a huge improvement over the TF-IDF. You'll notice it actually says metric equals Wasserstein in the bottom of that. Wasserstein is actually a supported distance metric in UMAP out of the box. The one catch here is that this was extremely expensive to compute. I'll discuss that in a minute, but just looking at it more, what we've done over the TF-IDF is completely separate out the sports stuff. It's all off in its own cluster. Same for the automobiles and motorcycles, the two different shades of green there, entirely separated out. We've really just completely refined everything. It's not perfect. There's still lots of fuzziness. There's still lots of misclassification. But if you actually look at the posts in the 20 newsgroup data set, they are very messy. So it's unsurprising that we're not perfect results. This is really, for this data set, a remarkable result. So really good results, slow performance. That UMAP run took literally hours and hours for a small number of data samples. And almost all of that time was due to the computational complexity of computing all those Wasserstein distances. So while we can produce a nice UMAP plot, it isn't that practical in general, which after all is probably what we really want. So are there any ways we can speed this up or make it generally more useful? Well, I mentioned a couple of approaches. The one I've been working on lately is working with linear optimal transport. And I've had a lot of success with this. So this is the one I'm gonna push here. I don't want to get into the details because once again, this is a whole field in and of itself. But the goal of this approach is to transform the data into a simple Euclidean space where the Euclidean distances in that new transformed space are actually good approximations of, or at the least, very strongly correlated with Wasserstein distances in the original space. It is a little computationally tricky, but orders of magnitude cheaper than computing all those Wasserstein distances, and it ideally not too lossy. And in practice, from what I've seen, it isn't. Really, all your I won't discuss the details. We can get to that if we have some time, and we can talk about that there.
No. Can you say the intuition sentence you were going to say?
So essentially what you want to do is you create a reference distribution, and you solve the optimal transport plan from that reference distribution to each data sample, which is a distribution in and of itself. And rather than taking the distance, I actually you take the transport plan, and you essentially take the offset vectors of that plan, and just linearize those, flatten that out. And that is your linear optimal transport representation. That's the guts of it. The mathematical justifications for why that actually works and is sensible and is valid are a little deeper, but you can hopefully see there's actually nothing very complicated going on there. It's not some really deep, complicated computational thing, other than solving a whole bunch of optimal transport problems and using the transport plans rather than just the distances.
Interesting.
And there are some catches with this too. Word vectors live on an n-sphere, not in Euclidean space. And so when you're working with spherical data, things don't quite work the way you'd want. So you have to make some adjustments. I managed to, the theory for that is not especially challenging. You can work out how to do that. I do have publicly available code to do all of these things. I can share that with you later. I'll put a link in the chat at the end. I will caveat that it might be publicly available code, but it's very much beta code that we're still playing around with. It's our little research playground. So it'll work for your problems, but I wouldn't trust it as a production use for anything yet. And the sand may change under your feet a little bit if you're relying on it, because we're still developing the repository.
And
so,
what about the theory ideas? Are you going to be writing those up for consumption?
Which of linear optimal transport in general? Other people have fortunately already well delved that theory.
I see. So you're using established tricks.
I'm using established tricks here. The only thing I'm adding is dealing with the spherical data as opposed to pure Euclidean data. If you Google linear optimal transport, you will find plenty of good references. There was a paper that came out, I think, either just this year or last year, that has excellent detail on all the theory behind it. So here's the results that I get with linear optimal transport. And you can see it very closely matches the overall shape and structure that we got from the Wasserstein distance. In fact, it resolves actually some of the noise even better. That's partly because I'm busy compressing things down a little bit, which removes some of the noisiness in everything. And the other advantage is that linear optimal transport with this technique, it produces very high dimensional vectors. I use just an SVD on those to compress them down to sort of something in the much more in the word vector range, 100 to 200 dimensional. That's not so hard to arrange for. But what I get out is document vectors that look just like word vectors, but now their notion of similarity between these document vectors is very much the Wasserstein distance style notion of similarity because of this transform from the linear optimal transport. So I can take those vectors and push them through to all sorts of other machine learning tasks that you like, clustering or other downstream supervised learning tasks. All of these all work. And in fact, I've applied these compressed linear optimal transport vectors for the 20 news groups to the classification problem. Can we classify which news group it is? And the vectors that I got out of this technique that I described here actually produced better classification accuracy than fancier techniques like universal sentence encoder. So this is actually surprisingly powerful. And to kind of summarize here, this is the sort of qualitative UMAP views of various different approaches to this. So on the top left, we have our pure word count that we started with. If you apply the domain-specific transformations, TF-IDF, you get the top right. The linear optimal transport results are on the bottom left. So that was using just the things that I have described so far in this talk. So just TF-IDF transformed bag of words documents, the very simplistic word vector training that I described, really just counting up co-occurrences, and then some basic matrix transformations, and linear optimal transport. And in the bottom right is Google's 1 GB universal sentence encoder deep neural network. And you can see why universal sentence encoder is, relatively speaking, very popular. I mean, compared to TF-IDF, which was the best that people had for a long time, it's far, far better at separating everything out and demonstrating all these much clearer categories and resolving a lot of finer structure. But I would argue the optimal transport result is at least as good as universal sentence encoder, and it doesn't require all this complex pre-training or dealing with the fact that it only really works for language. I think these techniques are entirely translatable to other problem areas. So just to kind of round things out, what are the takeaway messages here? Well, I would say NLP has done amazing things in recent years. And while those specific breakthrough models are usually huge black box deep learning models specifically designed for text, there is a lot of core ideas that powered these things that we can extract and build upon. And those parts are much more easily generalizable to other problem domains. The similarity or distance in the column space was the critical win here. This ability to have word vectors is the thing that really powered a lot of it. But all you need for that is locality or co-occurrence, locality and co-occurrence to sort of make sense of these things and build this column space distance that we can work with. And really, simple tricks like column space embeddings and linear optimal transport can compete quite favorably with massive deep neural network techniques designed specifically for text. So that's all I have. Very much, and hopefully we can try and discuss how the single cell domain in a little more detail. Thank you.